So my name is Aurelien Pelisiser. I'm going to present the work I have been doing in the IBM research in collaboration with ETH Zurich. So cell type specific regulation, gene regulation in rheumatoid arthritis. So this talk is focused on rheumatoid arthritis, but the method I will present here can be applicable to any other kind of disease. So just to make it very quick, rheumatoid arthritis is an autoimmune disease that is affecting around 1% of the population. So it's a major issue currently. And so what happened is that your bone cartilage is gonna swollen, caused by the autoimmune reaction of your body. So the motivation of this talk is in the current global landscape of RNA sequencing and single-cell RNAs. There have been a lot of progress in the gene regulation inference thanks to gene-wide association studies or differential gene expression analysis. We have led to the discovery of many novel therapeutics, but all of these methods generally they disregard information about the cell type specific gene regulation, and this is a major drawback because depending on the cell type, the gene regulation are going to be very different. So this is the information that we would like to have in order to design more specific treatments. So in this talk, I will address this issue, but so the outline will be at first I will just show you the synovial tissue in the context of rheumatoid arthritis, how the cell type, how different cellular composition we can have. And then I will show you a method to identify key driver regulating rheumatoid arthritis. And finally, I will show you how we can also infer our own regulatory network in a specific cell type. So just to show you a quick overview of the dataset I'm working on. So I'm working on public dataset that are already published. So on the left, it's a very big study with more than 200 patients with 152 rheumatoid arthritis patients and also around 30 control. But on the right, I'm using the data from the Accelerating Medicine Partnership, phase 1, which has the particularity of doing single-cell data in synovial tissues. So this data, I will come back to it later. This is the one we will use in our analysis. So first, the cellular composition in synovial tissue to infer that from the, so there is some tools to infer from the gene expression alone. You can try to infer the cellular composition. So it's not always very reliable, but it can be interesting to see what we can get. So for that, I'm using a tool called XCell, which was pre-trained on 64 cell type. So it's available in the study there. And this is what we get. So here it's very clear that depending of if this is RA tissue or healthy tissue, we can have major differences for some cell type, because this is expected because this is autoimmune disease. So the joints are swollen and there is more immune cells because of the autoimmune reaction. But you can observe here some difference, significant difference in B cells, in T cells, in monocyte, pericyte. So this tells us that the gene expression matrix that we had this slide here is heavily biased by the cellular composition. And this is not very useful for our cell type specific regulation, because if you do a differential expression analysis on this, you won't know if the difference will come from the difference in cellular composition or a true difference in the gene regulation in a specific cell type. So this was just to show you an overview of how the synovial tissue looks like. So how I'm going to address this issue today is I will present two independent methods, because it's nice to have independent methods, so then we can combine them, and it's make our analysis more reliable. So I will be combining differential gene expression analysis with a lot of prior knowledge from literature, and also some gene regulatory network previously published in literature, For example, the string database for protein-protein interaction, or also Bayesian network from Giant. So the first method is using a tool which is called merge omic. And the idea is quite simple. So as an input, you take two things. First, you need a list of genes associated to a disease. And second, you need a network. These two things. Once you have that, you can check each hub in your network. You can check how many genes associated to the disease the node is associated. And if the association is more than you expect by chance, then this node can be considered as a key driver. So now the question is, what do I use for the list and what do I use for the network? So for the list, because each study they can have different biases, so it was better to combine different studies and do some meta-analysis. So for the differential expression in the synovial tissue, I was checking the genes that are found consistently across different studies. And the same, I also made a list from literature where for different databases, so for example, the list of the target of the drugs that are already on the market, or list from the gene-wide association studies. So if a gene is found in at least two of these databases, then they are added to the list. These two, differential gene expression and literature, I take them as separate lists because it will make my analysis more robust if I have two independent lists. And for the network, I'm using the Giant database, which has the particularity of having cell type specific network. So, and also tissue specific. So for example, I have a network for NKT cell, for dendritic cell, for T cells. Voila, so this is what I have, what I'm using. And now what do we get? We get this. So let's spend some time on this plot. On the left, the original list, this is the two initial lists that I had at the beginning, the differential expression list and the literature list. And you can see here there is very low overlap between the two. But after I run the key driver analysis, the overlap becomes very significant, like more than half in most of the cell type. This tells us that even though the original list was quite different, the key drivers that were inferred are similar. And another thing you can notice here is the number of key driver is different depending on the cell type. So you have some cell type like dendritic cell or natural killer T cells that have a lot of key drivers. And we know that RA is autoimmune disease, so we expect, of course, T cell and B cell to be involved, but there is some other few cell types here, like eosinophils, that have not been described a lot in literature. So we expect this to be maybe interesting for experimentalists to analyze. So this was for the first method. So the first method doesn't really tell us details about the regulation. Let's say it can give you a list of genes, but it doesn't tell you these genes, what it is doing or what is it related to. So to answer this question, we need to infer our own regulatory network. So we have much more power much more prediction power regarding some regulatory information. So for this part, I'm making my own network. And to do that, I'm using a method called PANDA, passing message between biological networks. And this method is using three information that input. The first two, they are from prior knowledge, so transcripting factor interaction from the string database or the gene motif database. So the list of the target for each TF. And then the third one is the gene expression matrix from public data. So this network has a particularity to be a bipartite graph because each TF is related to a list of its target gene, and each arrows, they represent the likelihood of the interaction from the TF to this gene. And then what do I do? This is the key aspect here, is that I make with a method called Lioness. I make a sample-specific gene regulatory network. So it means here, you can see on the right, in this example, I have 5 samples. 3 of them are from the disease group, and 2 of them are from the control. And the networks are the same for all the samples, but the only thing that changed is the edge weight. And then you can do differential analysis, not only on the gene expression, but also on the edge weight. And this can tell us information. For each TF, we can check if there is any correlation between the expression and the edge weight. And this can tell us which TF are the most involved in the disease. So if a TF has a very high correlation between its differential targeting and its differential edge weight, we can expect that this TF might be very important for the disease. So here's an example here where, so this is how it looks like. So because the TF has around 1,000 targets, so each point here is one target gene. And if you can see a positive correlation between the weight score and the expression score. So this, I reproduce the analysis for each cell type. So remember the dataset I talked to you about at the beginning. I have a cell type specific gene expression matrix for 4 cell types, so monocyte, fibroblast, B cell, and T cells. So this way I can have a regulatory network and make a complete analysis for each cell type. And interestingly, the correlation between the regulation in each cell type is very low. So this graph shows you that confirms that what I was saying at the beginning. The regulatory interaction is very different from one cell type to another. So this is what we obtain at the end, like a list of, we obtain a list, let's say, by combining the 2 methods that I described, we obtain a list of promising transcripting factors. And here you can see that most of them were already known in literature, which kind of confirms the reliability of our analysis. But it can be interesting for us to explore the ones that are less known in literature. Just to conclude on the last slide is just to show you on the left that there is a high correlation, a good agreement also between the 2 methods. So I take the top gene from the first method and the top gene from the second method, and I check if they tends to have a higher score than the other. So this shows that it was significant. And the last thing that can be also interesting for experimentalists is to know if I give him a target, I said, this gene is important for this cell type, but he also needs to know if there might be some collateral damage, because you want to make a treatment that is specific to a cell type. You don't want this treatment to also affect other cell type. So this graph is interesting. For example, you can see FOST-L1 is highly expressed in fibroblast, but mostly not expressed in other cell types. So this one can be a good potential candidate for targeted treatment. So to conclude, I just show you 2 independent methods to do cell type specific gene regulation. And of course, the next step is to test this one in vitro. And of course, we can also do that for other disease. So this project was made possible thanks to my supervisor at IBM Research, Maria Rodriguez Martinez, but also to our collaborator who is a rheumatologist who will do the in vitro experiment. This is already under the way. And I would like to thank you for your attention, and I will be happy to answer any question.
Any questions from audience?
So annoying.
Hi, can you hear me?
Yes, I can hear you.
So thanks for the interesting talk. We know that this rheumatoid, this disease is very complicated, but I saw your slides about estimating over thousands of the key drivers genes, but in the end, how many genes are really essential or like key driver genes do we expect to be for this disease?
So this is a very hard question, because for this kind of disease, we actually don't really know the answer, and one gene can cause a cascade of many other genes. And this kind of methods, actually, what you have to do, you have to define some kind of threshold, like let's say p equals 0.005. So this remains a bit arbitrary. And at the end, in my list, you could see only a few of them because I combined the two methods together and I took the overlap. But in both cases, I had to define a threshold that I defined arbitrarily. So practically for an experimentalist, you shouldn't send him more than 100 genes, right? Because he cannot test all of them. But I don't have an answer to tell you only 10 genes are specific for the disease. It's very hard to know from a computational analysis.
Yeah, thank you.
Thanks for your talk. I have a question about the transcription factors. So from your networks, you have a list of a lot of transcription factors. Do you have any ideas how they interact with each other as a combination to regulate the disease?
Yes, that's a very good question. So actually, once you have these networks, this is another thing you can explore. You can explore the co-regulation between different transcription factors. And I didn't show it here, but actually you can cluster transcription factor together depending on if they tend to have similar edge weight to the same genes, then you say they are co-regulated. And on the other hand, you can see some transcription factors are doing opposite, like one of them is upregulating the gene, the other one is downregulating the gene, which in this case would be like opposite regulation. So this is also the one thing you can do from the gene regulatory analysis.
Any other questions? Okay, so when you do differential expression analysis and then make the transcription factor and gene networks, so how do you separate the up and down regulated genes? Like, do you consider them in single?
So in this, for this analysis, we want to know which genes are important. So it could be downregulated or upregulated. It doesn't really matter for this analysis because it means something is happening in both cases. So we keep them both. But for the biologist who wants to make the experiment, they need to know this because if they want to study the effect of a gene, they want to make a knockdown, for example, or they could make a upregulation, stimulation of the gene, right? So let's say I don't do it directly in my analysis, but it's still something in the information that we have to keep in mind for the experimentalists who want to make the experiment.